Welcome to Broad Eye, the podcast that explores knowledge gaps in ophthalmology and eye care. Hello, everyone, and welcome to another episode of the Broad Eye podcast. My name is Sean Maloney, and today I will be interviewing our guest, Dr. Stephen Redenti. Stephen is a professor of Biochemistry and Molecular Biology at the City University of New York. Stephen, welcome to the show.
Hi, thanks for having me.
I'm going to call you Stephen, if that's all right, because that's how I knew you back in 2007 when we first met back at a research lab. We were both working at Harvard. So maybe we could use that as a bit of a segue into just you giving a bit of an overview of your background and how you came to be a researcher and professor at the City University of New York.
Sure. Yes. So it's great to be speaking with you again years later, Sean. We worked together at Harvard at the Scapin's Eye Research Institute with Dr. Michael Young, and I remember we shared a lab bench, and I think we made some good discoveries together. I started when I was younger as a visual artist, studying sculpture and painting, and I did that for a few years. And closer to 30, I went back to graduate school for neuroscience at the City University of New York at Hunter College. And I focused on retinal biochemistry, in particular the electrical and biochemical properties of photoreceptors as they connect to the first synapse with the bipolar cells in the retina. And I worked with a nice professor named Dr. Richard Chappell, who was a former nuclear submarine engineer. Then I was also volunteering at the same time with an organization called Recording for the Blind and Dyslexic. And just by coincidence, I think I was moving into the field of visual research, or research to restore visual function caused by blinding diseases, perhaps. When I left the City University of New York is when I went to Harvard, and where I met you, Sean, and I spent 3 years there working between Dr. Young, and Dr. Bob Langer at MIT. And my goal there was to engineer retinal tissue using biodegradable and some biomimetic polymers. So these biomimetic means that we try to model these small substrates after the sort of elasticity and the texture and the architecture of endogenous retina. And after I finished my postdoc with Dr. Young, I was hired as an assistant professor here at the City University of New York, and started working on problems that I thought would be most important to move the field of photoreceptor transplantation forward, namely enhancing the migratory potential of transplantable photoreceptor cells, amongst other projects. And that it's been a 12-year journey. I started with some real original questions, and some curiosity-driven questions, and spoke to a bunch of old scientists when I was starting my lab, and that was their advice, to follow the research that piqued your curiosity. And now here I am, speaking to you today, and
fair enough. No, thanks for that overview. I completely forgot, but I knew this when we met, but that you had the visual arts and sculpting background. Is that something that you've continued as a hobby over the years at all?
I've kind of applied that creativity to my research, my design of experimental designs, and reaching into the field for the latest technology or creative ways to approach problems across disciplines. I think of my work as a scientist as a creative work in process. So
that's fair. That's fair. It echoes. We had a guest on the podcast in the past named Walter Wittich, who also had he's from Montreal. He's a professor focusing on low vision, and he had a very creative arts background as well, and then ended up in the I think he was neuroscience as well, and then into ophthalmology research. So it's there's probably some overlap not some overlap, but there's probably some benefit from that, developing the right side of the brain, and in parallel with the left, I guess. If you, I don't know if it's quite that simple. You're the neuroscience researcher, but anyhow, I'll dive into some more of the questions. I could talk about this stuff forever. So one thing that's fascinating me about your research, and you're talking about, cross-disciplinary insights and focus, is that you don't just focus on, the retina and biochemistry of vision, but your research also spans other domains like oncology. So, why is that? Is that to try to gain insights and apply that you learn somewhere and apply them to another domain, or what has been the impetus behind that, and if there's any learnings from one field that you've applied to the other?
That's a great question, and what I've done is I've worked with some systems pharmacologists and geneticists to identify mechanisms that are conserved between metastatic cancer cells and migrating transplanted stem cells, meaning a transplanted stem cell has to migrate, in particular in the retina. A transplanted photoreceptor replacement cell has to invade the retinal tissue and then take up residence in the host tissue. So there are a number of mechanisms that a cell uses to do that. There's migratory machinery. And as I started my lab and started looking at databases for mechanisms that drive cell migration, most of the databases are filled with cancer metastasis data, in particular the ingenuity pathway analysis database, which is probably one of the largest we work with. The majority of the gene expression data and the molecular data are all based on studies in cancer cells. So the parallel for me between transplantable photoreceptors and cancer cells is that they both use the same migratory machinery at different times. Obviously, metastasis in cancer cells is part of the pathogenesis you want to stop. So we take the mechanisms we find in the cancer cells and we try to upregulate them in our transplantable photoreceptors so that they will take on this aggressive migratory behavior or phenotype. That's one aspect. What we're studying is the cancer we're studying in particular now is retinoblastoma. And we are at the same time trying to knock down retinoblastoma genes to prevent it from metastasizing. And that's one line of research in the lab. But a parallel line of research in the lab is to take the genes we find upregulated in the metastatic retinoblastoma cells and try to turn them up in our transplantable photoreceptor cells to restore vision or to rebuild the architecture of the retina. Because as you may be aware, the success in the transplantation rate of new photoreceptors is very low. It's close to 1%. And that was one of the big questions I left Harvard with, is what can we modify in the individual transplantable cells that would increase their probability of entering a host retina and as a path to restoring vision.
That's, it's a kind of a creative, I guess a creative approach. And, back when I actually worked at McGill in an ocular pathology lab and doing, a little bit of ocular oncology research with ocular melanoma and retinoblastoma as well. And, some of these, invasion and migration assays were pretty standard. We were trying to downregulate that ability of these cells to, invade or migrate, and just using these, these in vitro assays. But I never really thought about the opposite, which, as you say, it seems so obvious in hindsight, but why would we not have tried to, I guess we knew less about the cells and maybe what was causing them to migrate at that time. But I think, is it the same things, like a lot of these MMP proteins and whatnot? I mean, I'm talking from my memory from 10 or 12 years ago, so I'm sure there's a lot has changed, but is there a certain class of molecules that really seems to govern this process?
We're looking at one example would be chemotactic guidance, which would be like a stromal derived factor and a CXCR12 receptor, but also the MMPs are involved. The same canonical machinery that you were probably studying is what we're applying in a novel way to enhance the migration of transplantable photoreceptor replacement cells. It's just not been studied in this context.
No, I think it makes sense when you're studying retinoblastoma and cells that would be most like the cells in the retina that you are trying to transplant it. It just seems logical, right? It just seems logical. It's like, why hasn't somebody done this before? But I guess those are probably the best projects too, right? They just don't. It's like, yeah, it just seems like an obvious thing to do. And it would be, what's nice, it would be, I would imagine, quite a database of information out there. Maybe not so much for retinoblastoma, but for tumors in general, right? Where a lot of the, like you said, the machinery might be similar. So probably being tapping into that database of information is probably quite valuable. So on your, I'm going to jump ahead on some questions here a little bit. I'm going to be a little bit all over the place, but I was looking up what your lab's focus is, and this probably ties into what you've just been talking about. But I probably don't remember this exactly as it was written, but it was something to do with how cells respond to like morphogenetic fields of biochemical and electrical signals, and how that might influence development of neural tissues. So could you just unpack that a little bit for us and maybe describe what that even means? what that means for, well, you said you teach undergraduate students, right? So if I'm an undergraduate student sitting in front of you, maybe if you had to describe what that means, could you unpack that for us?
Yeah, that's a fine way to put it. Yeah, I wrote this 12 years ago when I was probably trying to say a lot with, I had a lot of ideas. But to put it simply, we use this idea of a morphogenetic field to define the chemical, the tension, and the electrical signals present in the retinal environment in relation to the response properties of transplanted cells. So to explain it clearly, if I have a transplantable photoreceptor replacement cell, these are typically photoreceptor precursor cells. They're postmitotic, but they have a personality of their own. They have a set of sense receptors or a set of chemotactic receptors that are expressed on their surface. They also have the actin and migratory machinery of a newly postmitotic cell. So I call these the response properties of the cell. What can the cell detect? And you'll be surprised how much a single cell can respond to. And I'm always conveying to the students that you want to think about an individual neuron or an individual neural progenitor or a photoreceptor precursor as having a personality the way a young child would have a personality. The cells, for example, will respond to light. There's phototaxis. These cells will also respond to electricity. There is electrotaxis. Obviously, there's chemotaxis, which is that this is what we've studied the most of, is there are a set of chemotactic receptors expressed on the surface of these cells, and the retina is releasing fields of proteins that will bind to those receptors. And the cells will then make a decision whether to move forward, to move backward, or not to move at all in response to the field of ligand gradients coming from the retina, as well as the endogenous electrical field of the retina, and perhaps the softness or the hardness of the retina itself. So this is,
Okay.
How's that?
No, sorry, no, it's good. I was just gonna jump in with a thought, so which is probably makes no sense, but we'll try. You're talking about the looking at ways to influence the migration and integration of these photoreceptor cells or photoreceptor precursor cells into the retina when they're transplanted, because they don't have a very high affinity, I guess, in the first place. Has anybody looked at the opposite too? Like you're talking about the retina releasing these ligands into the environment and whatnot. Has anybody looked at how do we make the remaining retina as more permissible before we even transplant these cells? Is that something that's been looked at?
Yeah, it's been done in a number of studies. For example, there's a Mueller glial cell, as you know, that can become active and inflamed during disease and injury. And some studies have tried to break apart the Mueller cells to allow for the migration of newly transplanted cells into the retina. Others have added growth factors to the retina. And I think that some progress was made in those studies. But yeah, I saw room for understanding the response properties of the transplanted cells in relation to those other studies. And maybe these kinds of techniques can be used in parallel to optimize transplantation. Some pre-treatment of the retina with some optimization of the migratory capacity of the transplanted cells.
No, okay, that makes sense. So it's something I haven't heard of, and again, I've been out of touch with the research for quite some time, but it just seemed to, kind of make the soil as permissive as possible and make the seed, I guess, as best it can, and blast as many in there as you can, and try to optimize that procedure. What in the I like to talk about this stuff, the biochemistry vision, because a lot of what the general public is going to hear is clinical stage results from research, for example, and okay, there's a drug and it works, or a drug and it doesn't work, etc. But all of those discoveries are all ultimately based on or evolve from basic science research, right? It all has to start somewhere. It doesn't just start with a magical drug with a magical target. This is all stuff that's discovered in basic research labs like yours that then inform clinical stage type of applications. So I'm wondering if maybe you could, if anything comes to mind, over the last 5-10 years, I'll fill in that gap, like 12 years of my life. What are some of the more exciting discoveries or advancements on the basic science or biochemistry side of vision, and if any of those are still hot topics today?
Yeah, sure. Yeah, that was one of your initial questions. And so one aspect of the work we're describing in terms of potential new targets for therapies or potential new mechanisms that may be involved in the pathogenesis of retinal disease or the pathogenesis of ocular trauma would be what are extracellular vesicles. These are retinal extracellular vesicles, but extracellular vesicles are released from all cell types studied. This is kind of a decade-old field, but it's quite compelling now and drawing a lot of attention. I started doing this research 10 years ago. I didn't think it would become as exciting to everyone as it is, but these extracellular vesicles released from all cell types in the retina so far that we've studied contain DNA and RNA and proteins, and also mitochondria and some organelles. But the small extracellular vesicles are a signature of the cells that they're released from. So if the retina is under a state of disease, for example, something like age-related macular degeneration, then the retinal pigment epithelial cells in one of our recent studies is going to be releasing these extracellular vesicles that contain disease molecular characteristics and proteins. And then these extracellular vesicles can target cells within the retina that we believe drive the pathogenesis of the disease. Something like drusen, for example, is associated with exosomes or extracellular vesicles as far as we've studied them so far. So it also in something like retinoblastoma, we have studied the release of extracellular vesicles from these tumor cells, and this is really newly emerging data, but these cancer exosomes can target glial cells in the retina and then enhance the pathogenic environment to facilitate tumor growth. But this is not unique to the eye. The explosion of extracellular vesicle research is happening in all areas of biomedical research, in cancer as well as regenerative medicine.
How do you study that though? I mean, when you're like, how do you study these extracellular vesicles and their contents, given that it's not just protein or just, you know, DNA or RNA?
One step at a time, you know, they're
Oh, there's no magic bullet here. Okay, sorry. Yeah, it just seems complicated, right? To actually study these and identify signatures, I guess. I mean, on a high level, like what does somebody do to look at that?
Well, one of the first studies we've done is, first you have to allow the neural cells or the retinal cells to condition the media in vitro, then pull out the conditioned media that's filled with billions of tiny extracellular vesicles. These are about 100 nanometers in diameter, and they have a lipid bilayer, so they can protect the DNA and the RNA inside of them. And then you have to put them through an ultracentrifugation stage, which is just applying a very high amount of force for a significant amount of time, and you wind up with a tiny, barely visible pellet that is your extracellular vesicle pellet. Then you could resuspend those in media, and you can do RNA sequencing on them, which we've done, to get the full genetic expression of their cargo, their RNA cargo or their microRNA cargo. You can also do proteomic analysis, which we've done, which will give you a profile of the protein signature inside these vesicles. And then you can add them to cells. You can try to knock out genes and see if the genes are knocked out in the exosomes and if that changes the disease phenotype. But at this stage, we've been doing a lot of characterization, as you could imagine, because there's so much information inside the vesicles. We're just really at the tip of the iceberg, so to speak.
Now, I think is it generally thought that these extracellular vesicles contain like metabolic waste or just stuff the cell doesn't need anymore, or is it random? Like I'm interested. I find this.
Yeah.
I'm going to dive into this a little bit, if that's okay.
You know, when I started doing this research 10 years ago, I asked a couple of senior scientists if they thought, it was just molecular waste or if it was something significant. And they told me time would tell. And so there is selective packaging of proteins and RNA and DNA into these extracellular vesicles. So it's not waste, as you would imagine a cell removing metabolic waste through exocytosis. There's selective packaging of cargo, and that cargo has particular functions depending on the state of the cell that's releasing. So one advantage to this is there's an emerging field of biomarker discovery, because once cells release their extracellular vesicles, they can travel systemically and be collected from all bodily fluids. So with cancer, this is really coming out quickly, or something like bladder cancer. There's the collection of these exosomes or these extracellular vesicles, and they will really be able to give early diagnosis before a disease phenotype might be visible through observation or standard clinical exam. The specific packaging of cargo from disease cells, if it can be detected with a sensitive enough detection measure, could really advance early detection of, say, cancers and neurodegenerative diseases. This work is really gaining ground in Alzheimer's disease and Parkinson's disease and multiple types of cancers.
Oh, it's interesting. Like it's like you're talking about having cells in culture, and then you get the conditioned media. I guess back when I was a grad student postdoc, we just assumed that whatever was in the media, it's just cells have released that, and that's great. We never really thought about things being packaged in these extracellular vesicles, and that actually to access that you had to, centrifuge and disassociate these vesicles. So no, it's Pretty cool stuff. So you have a paper that you were involved in, I think it was 2019, I forget, but it was talking about these extracellular vesicles and drusen formation, and how these RP cells were releasing these extracellular vesicles. And I think you were trying to, reproduce something in vitro. Does that ring a bell, or am I off in left field on how I remember that?
Well, those are a couple of papers combined.
Okay, well, there you go. So you read more than one. But maybe you could just talk about that process a little bit, maybe in terms of, I guess, what you've learned from, from RP cells and extracellular vesicles and drusen formation, et cetera.
Yeah. So the first paper in ILVS a year or so ago was we pulled the neural retina out of a mouse model and collected the extracellular vesicles from that in vitro, just for a proof of principle, because we had not collected extracellular vesicles from retina at that point. And from that, we found several hundred proteins packaged inside the extracellular vesicles of that were released from the neural retina. That paper was a pure characterization, and we also sort of demonstrated the transfer of molecular cargo from the retina to target retinal cells at a distance using an in vitro assay. So that was to show that the release and transfer of molecular cargo was feasible and happening at a constitutively inside the neural retina. And then in a more recent paper, we were looking at the release of extracellular vesicles from retinal pigment epithelium derived from human endosomed pluripotent stem cell retinal organoids, which I'm working with in collaboration with Dr. Valeria Canto-Solar at University of Colorado, and the scientist taking the lead on that is Dr. Miguel Flores Belder, and he and I isolated together the extracellular vesicles from a disease model, an AMD disease model, using these retinal pigment epithelium in vitro, and we did this proteomic characterization of those extracellular vesicles and found that there was drusen associated with those extracellular vesicles, and that in a modeled stress situation in vitro, that the number of extracellular vesicles released would increase, and that would increase the amount of drusen as well. So those are the two studies, and these are initial findings. The paper, this paper is under submission right now for the retinal pigment epithelium work.
Nice. So we're going to publish this podcast before the journal publishes the paper, and then the Broad Eye podcast community, because I know about it before the general ophthalmology community. So no,
you're on the cutting edge here, Sean.
There you go. We try. So just, and again, this is just for my own curiosity. When we're talking about these extracellular vesicles being packaged and released from cells, what makes it so that they can be taken up or actually signal something on a target cell? Like, it's not like just a protein that's binding to a receptor on a cell, right? These are vesicles that contain proteins and nucleic acids and whatnot, right? So what do we know for certain that these things then, are taken up by the cells, or are they just signaling something extracellularly, or is that not known?
Oh, it's known. There's a large body of data on this. I think there were 3,000 papers published last year on extracellular vesicles. The field is exploding, as I mentioned. So these are internalized. Sometimes they're internalized by mechanisms of standard endocytosis. Sometimes it's receptor-mediated internalization. Sometimes the extracellular vesicle can fuse to the outer leaflet of a cell and just deposit its cargo into the cell cytoplasm. So there are multiple mechanisms, and it seems to depend on the condition, the microenvironment condition, or the condition of the releasing cell. The releasing cell then, outside of the lipid bilayer of the exosome, there's typically a cage of proteins. Sometimes there are receptors embedded in the lipid bilayer of the exosome, But on top of that, there is typically a cage of proteins that are attached to the outside surface of the exosome. So there's an interplay. There's protein to receptor binding driving this intake and other mechanisms of intake. But when the exosomes are internalized or extracellular vesicles are internalized, the cargo can be distributed throughout the cytoplasm, or it's often targeted to the perinuclear region, to the site of the endoplasmic reticulum, where a lot of protein synthesis occurs. RNA cargo shuttling is also occurring here. But if you look at it's really fascinating. If you look at we've done live imaging of extracellular vesicle internalization and the transport of these vesicles inside of a single neuron over time. They really fill the neuron and they get transported back and forth to the periphery of the cell and back to the nucleus on microtubule highways. They're being transported by motor molecules inside the cell. A lot of this work remains to be done to really define the destination of cargo from exosomes or extracellular vesicles inside of neurons. And a more comprehensive picture is yet to emerge. But
no, it's fair. Like I've missed this whole field in the last 10 years. Your papers are the only ones I've looked at that have anything to do with this. So apparently, I have another 2,198 papers just from last year alone to to refresh myself on this. But just before I go on to another question, I just wanted to circle back to you're saying that these RPE cells, whenever they are stressed in some way, that they were releasing more of these extracellular vesicles. Is the signature then of what's the cargo inside these vesicles different, which would then indicate a disease state if you're able to, like in the cancer, trying to detect this in people who have early stage cancers? Or is it just that there's more of them being released, or that could be disease specific, cell specific, or nobody really knows? There's a lot of ors in there.
Yeah, that's a perfect scientific question. The postdoc at Harvard. The answer is yes. The signature of a disease cell is imprinted on the exosomes it's releasing. So not only would there be more, but there would be more disease state cargo inside the extracellular vesicles. And the function then can be to facilitate disease progression. And that seems to be the case. I mean, in cancers, for example, it's very well established that exosomes released from tumor cells, whether it be glioblastoma or other types of lymphomas, when the original tumor site releases exosomes, these can enter systemic circulation, and where they land, they set up pre-metastatic niches, they're called. And the pre-metastatic niches then will signal to metastatic cancer cells to take up root and form new tumors in those areas that have been primed, or what's called educated by the initial tumor's cancer exosomes. It's really fascinating, the connectivity and the communication that these little vesicles facilitate.
It makes sense, right? Because when you think in cancer biology, we often think, okay, you have a very small cluster of tumor cells in an organ, and sometimes they remain dormant, I guess you can say, for a while, but during that time there was a thought that maybe they're kind of conditioning the tissue around them so that it is more permissible to grow and to essentially explode, right? But the idea that they could be releasing these extracellular vesicles from a distance to condition another target organ, for metastatic disease, is wild. And I'm sure that's a target of therapies in these conditions, right? Okay, how do we block these extracellular vesicles from even being released? Like, I don't know if I'm assuming that's some sort of target for a therapy.
Yes, there are many labs starting to research this area.
Fair enough. See, I'm so interested in the science, and I got into this stuff a long time ago. But I also like being able to ask people like you who have all the answers. fill me in on 1 decade in like 45 minutes, so it's good. It's very, very efficient. I just wanted to maybe cover one more topic, if that's all right. And it's just about the disease, the in vitro models. Like, so you mentioned, in the one paper how you had excised a retina from a mouse and looking at this in vitro, and then other one where you have taken human-induced pluripotent stem cells that you've essentially converted into RPE cells. What are some of the, I guess, benefits and limitations of using these, tissue-based, like in vitro models or cell-based models compared to, working with animals, which I guess is probably the next stage in some of these cases. But what are some of maybe the benefits and drawbacks?
Sure, perfect questions. So what I mentioned initially were human-induced pluripotent stem cell-derived retinal organoids, and I collaborate with Dr. Valerie Canto-Solar on these. We've been looking at the exosomes, but she is a world expert. And so a retinal organoid is really a three-dimensional retina with attached retinal pigment epithelium that is derived from human-induced pluripotent stem cells. And these
you can make this in a culture now?
Yes. For the past decade or so, it's amazing, Sean.
Yeah, this is it. All right, fair enough. I keep going. I'm educating here. That's good. That's good. No, that's cool. That's cool. Okay.
Since you and I were trying to generate retinal tissue back in Boston, this has really taken off. So these retinal organoids, they recapitulate spatio-temporally the main steps of retinal development that you see in vivo, and they form three-dimensional retinal cups with all the major neural cell types present and the retinal lamina highly organized. So there are also some really new, this technique remains to be standardized and optimized. At this point, it's valuable to use in conjunction with other models of drug discovery, like in vivo models or mouse models or cell models. But when it's standardized and can be efficiently and produced in a cost-friendly manner, I believe these will really be beneficial for drug discovery for retinal disease. They do have some early photoreceptor outer segment development, and they actually can detect light. So it's quite fascinating to look at these little retinal cups in a dish. The one other benefit of this is that they can be patient specific. So if someone has a single gene, something like retinitis pigmentosa, these skin cells can be derived, a retinal cup can be formed, different gene therapies can be targeted. And then there's a wide spectrum of diseases like Stargardt's disease and others that are these human-induced pluripotent stem cell optic cups are being generated, and drugs are being tested drugs and genetic modifications are being tested to bring the field forward. So those are the strengths, I think, to augment the existing drug development pipelines. It won't replace in vivo models, because I think the weakness of these retinal organoids, even though they're fantastic, is there's no immune response present in vitro with these models, and there is no vasculature present. So it's an amazing model for retinal development in vitro and retinal function, but it's lacking some of the major obstacles of effective drug therapy development in vivo.
Oh, that's fair. I think that it just still seems light years ahead of where it used to be, where we would just try to seed a monolayer of these harvested retinal progenitor cells or then induced pluripotent stem cells in a dish or something, right? So it just seems a little bit a big step closer toward in vivo, but still in vitro, right? That would probably yield more useful information. So this is interesting. This is you're making me want to get back into research. I mean, I'm still in science in some ways in my with the podcast and outside as well. But this is fun. I'm gonna stop drilling with your questions, and we can wrap up. I don't know if there's anything else specifically you wanted to highlight, but be careful if you do, because it could be a trap. So
you're still a scientist. You always will be, Sean.
Yeah, I know. I'm a lot of things. When scientist, I don't know if I'm still a scientist, but no. But I wanted to thank you for participating. I certainly appreciate you coming on and sharing this information with everybody listening. At some point in time, I'd love to sit down and have a coffee with you and learn what else I don't know from the last decade of retinal research and crash course. So sometime hopefully we can do that in person.
Absolutely. It'd be great.
Excellent. Dr. Stephen Redenti, thank you for joining me today.
Thank you, Sean. It's been a pleasure.
Right, take care.
Thanks.
And that concludes today's episode of the Broad Eye Podcast. If you enjoyed this episode, please be sure to subscribe on your favorite podcast platform. Of course, ratings and reviews are always welcome. And you can certainly share this episode with any of your colleagues or friends who might enjoy it. Thanks for listening.